Hey guys, this is Mr. O'Brien here, and what we're going to be talking about here is pretty much the fundamentals of genetics. So genetics, how traits pass from parent to offspring, things along those lines. So genetics is a field of biology devoted to understanding how characteristics are transmitted to the offspring. So how are these genes going from parent to offspring? Okay, and why are these, why are some traits more prevalent than other traits? So this is kind of what we're going to be talking about in this video. So the first guy we got to talk about is a guy named Gregor Mendel. Now, Gregor Mendel was a monk who studied heredity using pea plants, and he was pretty much what's known as the father of genetics. He was the first person to study it, and he was the first person to kind of show that our traits are passed in a mathematical way. The reason he was so good at proving genetics was he had a statistics basis, and he was very good with numbers and also recording numbers, and very tedious with his garden. So heredity, when we talk about heredity, we're just talking about passing of traits from parent to offspring. Okay? So how are these traits, how are these characteristics passed to our offspring? Okay? So why are your kids look the way they do? Okay? Now a trait that we talk about up here is a genetically determined variant of a characteristic. So is it round, wrinkled? Okay? Is it yellow or green or white, pod shape? All of these are different traits that pea plants had and that Gregor Mendel kind of started talking about. Now, Gregor Mendel, he had a procedure of how he tested his things. Now, the first thing he did is he got what's known as a P generation. Now, the P generation were our parents, and the P generation were what are known as true breeding. Okay? Now, true breeding is very important to this whole procedure. True breeding plants are ones that are either homozygous in nature. Now, they could be homozygous dominant or homozygous recessive. So what that means is they're either both capital letters, dominant, or both recessive letters, homozygous recessive. So he then breeded these two together. And if we know anything about genetics, when we breed two homozygous plants together, homozygous dominant with homozygous recessive, we're going to get all of the offspring having the dominant trait. So this is what's known as the F1 generation. It's pretty much the kids of the parents, the filial generation. Now what he did then to determine dominant and recessive is he then self-pollinated this F1 generation. And by self-pollinating, he knew that he was crossing all of the same genes. So when he did that F1 generation, he got 3/4 purple, 1/4 white. Now the 3/4 purple, those were the dominant traits, the ones that were showing up the most. The recessive traits, or the white is going to be recessive because it showed up least. So this is kind of how we discovered this law of dominance and recessiveness, by doing this process of taking the P generation of true breeding homozygous individuals, crossing them together, and then self-pollinating those to get the F2 to determine what we have. Now the law of segregation pretty much says that offspring acquire one factor from each parent. So pretty much offspring, they acquire one factor or allele from each parent. So pretty much, if you think about it, right, your mom has sex chromosomes XY, your dad has them XX. So here's the thing. When we talk about this, what we're talking about is you're going to get an X from your mom, and you can either get an X or a Y from your dad. So this is what's going to determine the sex of the individual. Now, this happens because you can only get one or the other. Offspring acquire one allele per each, one from your mom, one from your dad, and that's always how it's going to be. Gametes only contain one allele per trait. So the law of independent assortment, kind of the same thing, but it's talking about alleles separate independently of one another during the formation of gametes. So in this process, right, let's just say, you know, for example, we have capital G, lowercase g. When we form our gametes, the gametes are going to get one of the two. They're either going to get the dominant trait or they're going to get the recessive trait, okay? And this is kind of this law of independent assortment. They're going to split apart from each other. They're not going to be inherited together. And this is kind of what makes us so different from one each other. So the law of dominance and recessive, first thing we want to talk about is pretty much what an allele is. So an allele is, it's one of two alternatives alternative forms of a gene. So in other words, it's either the dominant or the recessive. And what does dominant recessive means? Dominant usually means it's going to be fully expressed. So the thing about a dominant trait is when it shows up, it's always going to be expressed. So for example, dominant alleles are expressed as a capital letter. Anytime you have a capital letter, that is the trait that's going to be expressed, even if there is another recessive one there. So recessive alleles are not expressed unless two copies are present. So this is distinguished by a lowercase letter. So here, this picture over here kind of represents, okay, the dominant trait is the one that is going to be the most dominant. It's going to take control. So when it's there, it's taking control. Recessive, they're very passive. The only time that they show up is when there's other recessive alleles with them. So this brings us to genotype and phenotype. So genotype is our genetic code. All right? And the way I always remember this is genetic, right? Kind of sounds the same. Our phenotype is our physical characteristics. Okay? So again, phenotype, physical characteristics. So I always like to explain this over here. Now, your genotype could be, you know, like here's two different genotypes. Actually, three different genotypes. They are the letters. They're the code. Genotype codes for the phenotype. So for here, dark is dominant to light. So if you see a capital letter, it's going to be dark. See, capital A, the recessive one is not expressed, it's going to be dark. Only time the recessive allele is expressed is when there are two copies of it present. Now, homozygous dominant, homozygous recessive, heterozygous, pretty much what this means, homozygous means two copies are the same. So homozygous dominant, we're going to use the letter B here. Homozygous dominant would be two capital alleles, or two dominant alleles. Homozygous recessive, two lowercase b's. And heterozygous is a mixture. Okay, one is capital, one is lowercase. Now, probability comes into play a lot when we talk about genetics. Probability is the likelihood of a specific event occurring. So probability is the number of favorable outcomes divided by the total number of outcomes. Okay, and that's how we get our number. Now, this can be expressed one of two ways. It could be expressed as a percentage, 30%. It can be expressed as a decimal, 0.3. Or it can be expressed as a fraction, like 3 over 2. So all of these are different ways to express probability when we're doing genetics. Now, how we determine the likelihood of an event occurring are doing these things called monohybrid crosses. And they are performed using this thing called a Punnett square. Now, monohybrid crosses only track one trait. So let's look at this. If green is dominant to yellow, what would the result of the two heterozygous green plants be? So we have two heterozygous green plants. So we're going to use the letter G. Heterozygous, one capital, one lowercase, and we have two of those. Okay? So when we do the cross, we put the letters, we kind of start on one. We start on here, and we meet in the middle between them. Okay? So what we're going to get here, we're going to get three different genotypes. One homozygous dominant, two heterozygous, and one homozygous recessive. Now when we do the phenotypic ratio, there's going to be 3 green, these three, and 1 that is yellow. Okay? So it leads us to a thing called a test cross. Now a test cross is pretty much done when we don't know what the genotype of something is. So the test cross, individual of an unknown genotype is crossed with a homozygous recessive. So we have this purple flower here. This purple flower, we know for a fact that it at least has one dominant allele because it is purple, because there's always going to be one there. So what we're going to do is we're going to cross it with a homozygous recessive individual, 2 small p's. Now the result of this cross is going to tell us what the genotype of the purple flower was. If they all end up being purple, we know that it was originally homozygous dominant. If it is white, if there is some white in it, we know that it was heterozygous for purple. So that's kind of what a test cross does. It looks at it, it crosses it by something with 2 recessive alleles to determine its genotype. So after doing these crosses, there are some different types of dominance that we have. Now, complete dominance is what we've been talking about. One is dominant to the other. Tall, dominant to short. Green, dominant to yellow. It's the normal one. But there's a couple weird ones that we have. And the first one we're going to look at is something called incomplete dominance. So in incomplete dominance, we have our homozygous dominant, that is codes for red. Our homozygous recessive codes for white. Now here's where it's a little bit different. When you have a heterozygous individual, it's no longer just coding for the red. It's a mixture of the 2 phenotypes. So the heterozygous is a mixture of the red and the white, and it gives us pink. Next one we're going to talk about is something called codominance. Now codominance does occur in blood typing as well. But in codominance, what we look at here is when we have a white one, it's capital W, a black one is capital B. When we get a mixture of the 2, both of those genes are expressed, so we get a speckled chicken. So codominance is pretty much when both traits are dominant and expressed at the same time. All right, so that's all I have for genetics. Hopefully this helps you out in the test, helps you out learning a little bit of genetics. This is Mr. O'Brien, signing off.